All right, so this section is going to be longer than you would probably like, okay? And then when I tell you to write some things down in the margins of the notes, just make sure that you do so. So that being said, this is about the cell cycle, which by definition, as it says at the top of the slide, it's the sequence of growth and division of a cell. And you often see a pie chart that looks like this associated with the cell cycle. Let me see, it looks a little fuzzy. Let me see if I can do something about that. Okay, so that being said, the cell cycle is divided into two main phases, interphase and the M phase. So when we look at the pie chart, interphase is that whole middle purple region, okay? And then the M phase is this little piece of the pie out here, all right? So those are the two main phases of the cell cycle. And then within the cell cycle, you have the other phases that you see there, okay? So then you've just got a nice little, flow chart down at the bottom, right? That the cell cycle is split into interphase and the M phase, and then interphase is split into the G1, S, and G2 phases. Notice that the G1 phase, that's where the cell is growing, it's getting ready to divide, as far as just making new molecules, new proteins, new organelles that are needed for division. And then during the S phase, that's when DNA is replicated or synthesized. So the S stands for synthesis here. Okay, so you might want to write that down. And then the G2 phase, this is when the cell's preparing for nuclear division. Essentially, organelles and molecules that are necessary for the cell to divide are going to be produced during the G2 phase. And G stands for gap. You don't have to know that specifically, but just kind of an FYI, okay? And then within the M phase, you've got two phases, mitosis and cytokinesis. And then mitosis is split into four phases: prophase, metaphase, anaphase, and telophase. Collectively, the M phase is cell division. Interphase is everything that's happening in order for the cell to get ready to divide during the M phase, okay? Hang on just a second. Right, so the phase that you typically start with the cell cycle is interphase, but it is not a beginning phase. Notice it says it's the period between successive rounds of division. All right, so there's three phases within interphase. I already mentioned them: cell growth. In the G1 phase, new proteins and organelles are going to be made. The DNA is going to be copied during the S phase, which stands for synthesis, or replicated, okay, duplicated, so that each new cell is going to have the same amount of genetic information as the parent cell. All right, so after the S phase, there are two identical copies of every strand of DNA in the cell. Then the nucleus is going to prepare to divide during the G2 phase. All right, so then the M phase. Within the M phase, there are two phases, mitosis and cytokinesis. So mitosis is specifically nuclear division. So notice that that's italicized. You might want to highlight that. You might want to make yourself a note that just reminds you that mitosis is only technically the division of the nucleus. Sometimes you hear it referred to with cell division, but it's really just nuclear division, okay? So after mitosis, there are two new nuclei that are genetically identical to the parent nucleus, okay? It's going to follow interphase, and it's split into four phases. And over here, what you're seeing is just an animation or a cartoon on the right of all of the phases of the cell cycle within a plant cell, and then you're seeing the same thing on the left, specifically in an onion root tip. So this is interphase, and then this is prophase, metaphase, anaphase, and telophase. And if possible, we're going to go to lab and look at cells under the microscope, and you identify them in each different phase of the cell cycle. That'll kind of depend on what's happening with our schedule. Okay, so let's go next to the phases of mitosis. So prophase is the first phase of mitosis, okay? Pro essentially meaning first in this case. So during this phase, and again, this is one of those sections of notes, or I said this at the beginning, that if you are listening to this on Monday, January 4th, and you haven't been to class yet, then some of the things I might say here, you're going to maybe be a little bit lost on, and that's okay, okay? So prophase, again, is the first phase of mitosis. So here, the DNA is going to coil into visible, replicated, or duplicated, or copied, we can use lots of different words there, chromosomes, and those look like X's that we see over here, Okay. Other things that are going to happen in prophase, so what are called centrioles. These are going to form at the poles of the cell. They're these little red kind of barrel-shaped structures that we see down here in this diagram. They're going to duplicate during the interphase, and then they're going to migrate to opposite poles of the cell during prophase. And they don't form in plant cells. They only form in animal cells. And then the spindle fibers, which are these things that kind of look like little fireworks down here, and up here, the spindle is going to form between those centrioles. And so then the replicated chromosomes are going to get organized, and they're going to attach to the spindle fibers. You don't really see evidence of that in this particular set of diagrams because the nuclear envelope is shown as a dotted line. So it's not completely broken down yet, but the nuclear envelope is going to break down, and then the nucleolus is going to disappear. Once that happens, then those X-shaped replicated chromosomes are going to attach to spindle fibers to get ready for the next phase. Okay, and real quickly, in this picture down here at the bottom right-hand corner, that says late prophase. Basically, those chromosomes are really dense, they're tightly coiled, they're all X-shaped. The nuclear envelope's gone, okay, and they're getting attached to spindle fibers to get ready for the next phase. All right, so the next phase is metaphase. Meta means middle in this case. So the replicated chromosomes, they are attached to the spindle fiber. So you can see the 4 different colored chromosomes, and that's just the way this particular set of diagrams shows them as 4 different colors. But you can see those lined up in the center of the cell there. Each one's attached to a spindle fiber. Okay, and there's a spindle fiber coming off from each replicated chromosome from each side, right? And then the spindle fibers are anchored at the centrioles that we see at the poles, right? So they're lined up so that then in anaphase, okay, when you put AN in front of something, or ANA in this case, it makes it mean not or the opposite. So this is where now the replicated chromosomes are going to break apart, right? And they are typically shown as a little V. And so now the sister chromatids, right, are going to move to opposite poles of the cell, right? And these are now called individual daughter chromosomes. So that being said, this is an individual daughter chromosome or an individual chromosome. There's a lot of ways we could say that. And so these were, for example, the 2 red ones that we see here, they were attached at the top, right up here. Sorry, my hand's totally in the way of everything else. Okay, so that's the replicated chromosome. So this is a replicated chromosome, and it is made or duplicated or copied, and it's made of 2 sister chromatids. Okay, and so I would write that down. Made of 2 sister chromatids. So those sister chromatids are identical, okay? Maybe make yourself a note of that as well, that those 2 sister chromatids are identical. So one sister chromatid is going to move to each pole when they split apart, okay? And so then they're going to be individual daughter chromosomes. And so this is happening because the spindle fibers are breaking down at the end that's closest to the pole, and as that takes place, it breaks the centromere that attaches the 2 sister chromatids to each other. That's in the notes that you're going to talk about in class. And I won't try to show that because it's a tiny little bitty picture. It's going to break that centromere apart as those spindle fibers break down. And it's enzymes that are causing that breakdown process to occur. Okay, and so then here we've got another diagram of this that's going to show a chromosome number of 8. Okay, this is showing a chromosome number of 4 moving to each pole. Some of that we'll talk more about eventually. This is in a plant cell, okay, anaphase in a plant cell, anaphase in another plant cell. Up here, you've got metaphase in an animal cell and metaphase in a plant cell. Looks kind of like a little spider. And then these, I always say, kind of look like you're pointing your fingers towards each other. Then the last phase is telophase. Okay, so this phase starts once the daughter chromosomes get to opposite poles of the cell, right? And so that being said, several things that are listed here. So the chromosomes are going to unwind. They're going to become much less dense. That'll make more sense once you've heard what we talk about in class on the first day if you're listening to this before then. The spindle fibers are going to break down. The nucleolus is going to reappear, and then the new nuclear envelope is going to reappear. So you see that as a dotted line there, okay? So now, each of these two nuclei are genetically identical to the parent nucleus. This is telophase in a plant cell. This is telophase in an animal cell, And telo, T-E-L-O, means the end, okay? So if you think of a telephone, right, and I know y'all aren't used to talking on a phone that's plugged into a wall, but think of the fact that if you were not in the same room with two people being in the same room on their phones, speaking of, that was mine, that you would be at the end, okay, of, like with a traditional phone, at the end of two different wires that are connected, okay? So telo means end. All right, and so then cytokinesis, okay? So cyto in this case is referring to the cytoplasm. And so kinesis, this is a cleaving of the cytoplasm, okay? So this is typically going to start at some point during telophase. So if you go back to that original pie chart, you'll notice that there's a little bit of overlap, like once telophase begins, right? And I actually think that on that diagram it shows it starting, I'm looking at it real quick, it kind of shows it starting where it may happen, or cytokinesis may begin in anaphase. We typically say it takes place and starts more during telophase, and that's where the cytoplasm is cut or cleaved in half. In animals, the plasma membrane is going to pinch in, and it's going to create what's called a cleavage furrow that we can see there in an actual cell dividing. This is a cleavage furrow forming. This is one forming here. So this is cytokinesis having started, just the pinching in of the cytoplasm. I always say, kind of think of this like you have a big dough ball, like bread dough, and that if you just kind of stretched it, that eventually as you pulled two chunks in your hands apart, that eventually that would break and you'd have two balls of dough. Okay, and then in plants, this cleavage furrow does not form. Instead, what forms is known as a cell plate. Okay, and that's, as you can see, pretty distinct structure that's in the cell. And on either side of that cell plate, a new cell wall is going to form to turn these two sides of this big rectangular cell into two new genetically identical daughter cells. Right, and we're working toward that in this diagram as well. Okay, so to go to the next slide, okay, and this is a set of diagrams that's out of a really old textbook that we used to have, and y'all don't have a textbook. So there's a few things that we're going to do in class to just make sure that some of the little subtle nuances about this hopefully will click a little bit better, because there's some things about diagrams that scientists don't typically show. So for example, interphase is typically shown where the nucleus is really modeled in appearance. Nice solid nuclear envelope. You see a set of centrioles and spindle fibers forming, but you can't see the DNA because it's not dense enough to appear as chromosomes that we see during prophase. And so then during prophase, the nuclear envelope breaks down, the chromosomes coil into visible X-shaped chromosomes, which I feel like that's really dark. I think maybe it's the lighting in the room. But you've got a red chromosome, a kind of yellowy orange chromosome, a green chromosome, and a blue chromosome. Okay, and so that being said, the chromosome number in this particular diagram is 4, and we'll talk about what that means soon too. Okay, so then that's prophase. Metaphase, the chromosomes line up in the center of the cell. Each one's attached to a spindle fiber on either side. Spindle fibers are anchored to the centrioles at the poles. Then those replicated chromosomes split apart during anaphase, and now each half of the replicated chromosome is called an individual daughter chromosome. And this half down here is going to move to this pole, and this half up here is going to move to that pole. Then cytokinesis is going to begin during telophase. Telophase begins the minute that the chromosomes reach the poles. They start to unwind. A new nuclear envelope forms around them. New nucleolus appears. The spindle fibers break down. So then when cytokinesis is completed, and that's what's being shown here, then that means that the cytoplasm is completely cut in half, and you have two genetically identical daughter cells that are just like the original parent cell. Okay, and so this is showing the beginning of cytokinesis. So that'd be a cleavage furrow. This would be an animal cell since it's nice and rounded. If it was a plant cell, it'd be real boxy and rectangular in shape. Okay, and so then not all diagrams show a separate telophase and cytokinesis being complete. Okay, some kind of could just combine it. So that being said, now these two new daughter cells can go through this process. If they both go through that process, then you're going to end up with 4 cells. And then those 4 cells could go through that process, and you would have 8 cells. Okay, hold on just a second. All right, and so that being said, at the top of this slide, it says together then mitosis and cytokinesis produce 2 identical daughter cells that are going to grow, carry out normal daily activities, do what they do as cells until they have to divide. Okay, so we could be talking about a single-celled organism like a paramecium or an amoeba, for example, or we could be talking about cells within a multicellular organism. Okay, so as an example, as you sit there and listen to these notes or wherever you are all day long, your skin cells are dividing really rapidly compared to some other cells in your body. So you constantly make new skin cells. Most of dust is dead skin. Okay, and so that being said, about every month you've got an entirely new layer of skin because your skin's constantly going through this process. Okay, so within your body, okay, the parent cell versus the daughter cells we've got to talk about in multicellular organisms with this term diploid here. There's another term that's haploid that we're going to talk about with that. Let me stop for just a second. All right, so everything that we're going to focus on where it says the parent cell and down is going to be focused on multicellular eukaryotic organisms, okay? So, and we're going to just couch this in terms of humans. So inside your body, most of your cells are what are known as diploid, okay? And they are known as somatic or body cells, right? Every diploid cell in a normal human, okay, or normal organism that's multicellular, I should say, every single one of those is going to have an even number of chromosomes, right? And so this says the parent cell's diploid, has an even number of chromosomes, and then the 2 daughter cells that are also diploid are going to have the same even number of chromosomes at the end of this process, right? And so, that being said, right, we're going to talk about another type of cell division that's meiosis, but we're not ready for that yet, okay? But we've already talked about eggs and sperm a little bit at the very beginning of the year, okay, as far as reproduction and some things, just to kind of introduce some basic characteristics of life. So when egg and sperm fuse, they make a single cell called a zygote, okay? Each egg cell and each sperm cell has, in the case, and so now we're looking down here, each of those has 23 chromosomes in it. When the egg cell with 23 chromosomes, and this is just for humans, sorry, I should have said that. When the egg cell has 23 chromosomes and the sperm cell has 23 chromosomes, 23 plus 23 gives you 46. Those fuse together to make a zygote. Okay, so that zygote is a single cell that's going to grow and divide by mitosis to turn each organism, in the case of this example with humans, into a new multicellular human. Okay, by definition, diploid di means 2, so this is a cell with 2 copies of each type of DNA. Right, the N in this case is the number of different types of DNA. So again, in humans, we have 23 chromosomes, right? So in all the cells of your body, your somatic cells, you have 2 copies of every chromosome. You got 1 copy from your mom, you got the other copy from your dad for your total of 46, okay? Mom gave egg, dad gave sperm, those fuse, you get a zygote, that zygote grows and divides by mitosis. And every new cell that's formed is going to have 46 chromosomes in it. Again, assuming it's a normal chromosome number. In this case, in humans, we could talk about some other chromosome numbers in other organisms, and we will at some point in time. Okay, so that being said, the last slide here. So this is about what is called a homologous pair of chromosomes, or homologous DNA strands, homologous chromosomes. Okay, so homo means the same. Okay, so homologous chromosomes are paired strands of DNA where you get 1 from each parent. So 1 comes in the egg cell, 1 in the sperm cell. The egg and the sperm fuse to make the zygote. So let's consider this picture down here of these 2 blue chromosomes, the zygote of some flowering plant, okay, a pea plant, like Mendel's pea plants. That's what this is supposed to be. Okay, so in the egg cell and the sperm cell, they each have a chromosome that's this length, okay, this shape and size with this particular gene and many others along its length in the same location. Okay, and so homologous chromosomes, they're paired, they're the same shape and size, they have the same genes for the same traits arranged in the same location or same order, but they are not identical to each other. Okay, and that's because they may have different possible alleles or forms for the gene. We're not really ready for that term yet, okay, but just know that that's coming. This picture is supposed to show the allele for purple flowers versus the allele for white flowers, but the coloration on those two kind of pinkish bands is kind of hard to tell. But that's a good example of how those would not be identical, but they're the same shape and size with the same genes in the same location. Right, and so this is a non-replicated homologous pair, so is this, and then this is showing a replicated homologous pair, right, with two sister chromatids in each replicated X-shaped chromosome. And then this is showing the same thing over here. So we'll pull back to these diagrams here. This is always a really confusing thing for a lot of students, okay? So more to come on that. All right, and so then the last two slides on this set of PowerPoint notes. This one here about regulation of the cell cycle. So notice it says cell growth and division are carefully controlled. Over here, we see a graph of what are called a bunch of different cyclins. Each colored line on there is a different cyclin. So think of these as proteins, as enzymes that are controlling the cell cycle. Some of them keep the cell cycle progressing along its way. Some of them inhibit progression, like if some phase is not finished yet, then that a cyclin will say, okay, like to the cell, kind of send a signal that tells the cell, all right, this needs to finish up before the cell can go into the next phase. Okay, so some progress it, some inhibit it. But just to kind of explain this with this particular picture. So it says cells can be put in a petri dish, and y'all are going to do this in the spring, where you're going to put bacterial cells in a petri dish and they're going to grow. But cells can be put in a petri dish, they're going to grow until they come into contact with each other, and that's what's happened here, okay? And so then this is couched in terms of an injury, right? Like this kid falling down and scraping his knee. So then if you fall down and scrape your knee, those cells are injured in that area, and then they are going to grow and divide really rapidly by mitosis to lay down new cells to repair that injury, right? And then the younger you are, the faster cell division is going to occur. So typically we talk about the fact that younger kids, you will scar more easily because you're going to turn over cells really fast, and you'll heal much more quickly, where for me, I might not scar, but it might take me longer to heal if I were to fall down and scrape my knee, and then it would take somebody who's 80, even longer to heal, okay, as far as having some sort of injury. So this is a good example of how you've got cells that have grown and divided until they touched each other. You have an injury that needs to be repaired, and they're going to grow and divide until they touch each other again to repair that damage. Okay, and then again, so here's at the bottom where it talks about proteins controlling the cell cycle, some inhibiting and some progressing it. And then that just talks about how that growth can be turned off and on. Okay, and I'm going to just mention this really quickly because you've got an assignment that you're going to do that's going to deal with cancer. By definition, cancer is simply uncontrolled cell growth and division, right? And cancerous growth produces masses of tissue that are called tumors. Tumors are a problem because they need a whole lot of extra nutrients and oxygen to function, and so they're going to deprive their non-cancerous neighboring cells of nutrients that are needed, and so they damage surrounding cells. Cancer is typically a problem that results with, or is the result of problems with enzymes that are controlling the cell cycle. Essentially, we're just going to kind of keep this kind of simple for right now, that cells don't respond correctly because of problems with enzymes here. Sometimes it's a failure to produce an enzyme. Maybe it's overproduction of an enzyme. Maybe it's production at the wrong time. There's a lot of factors that play a role in this. There's a lot of environmental factors that also play a role. So I want you to think about that, what some of those environmental factors are that we hear are cancer, causing factors, okay, that maybe we, can control, maybe some things we can't control. Genetics can play a role as well. And just here you can see these cells look normal. They have a large amount of cytoplasm, a single nucleus. The chromosomes look normal. Okay, the chromatin specifically is mentioned there. And then here, very little cytoplasm, lots of nuclei. The chromatin's really coarse and dense. Okay, large and multiple nucleoli, and a small nucleolus. We haven't talked about that structure enough for that to be super meaningful for you in that diagram. But then you've got cancerous cells shown there and there. Let me know if you have any questions.